In Unit 3, Lecture 1, Part 3, we're going to be looking at the role the ability to adhere to host cells and resist physical removal plays in a bacterium's ability to colonize the host. Once again, make sure you read your fundamental statements or bullet points for this SoftChalk lesson and learn the detailed learning objectives that you'll need to know for your Unit 3 exam. We have another highlighted bacterium in this SoftChalk lesson, Neisseria Meningitidis. So remember, our topic right now are virulence factors that promote bacterial colonization of the host, things bacteria do to establish themselves in the host. And we're going to look at this SoftChalk lesson at the second of these six different groups of virulence factors, the ability to adhere to host cells and resist physical removal. So one of the body's innate immune defenses is the ability to physically remove bacteria from the body. And this is done through a variety of means. We constantly shed the surface epithelial cells from both our skins and the mucous membrane to remove any microbes that might be attached to that outer layer of cells. We can remove bacteria by such physical means as coughing and sneezing in the terms of respiratory organisms, and vomiting and diarrhea in terms of gastrointestinal infections. And of course, all the bodily fluids, things like saliva, blood following an injury, mucus, and urine all have a flushing action to flush microorganisms out of the body. So for bacteria to colonize the body, they have to be able to resist this physical removal by innate immunity. And they do this through three means that we learned back in Unit 1, by producing pili, cell wall adhesins, and or biofilm producing capsules. So the first of the three means of attachment we learned about in Unit 1 are bacterial pili, or fimbria, another name for the short attachment pili. So remember, pili enable organisms to adhere to receptors on target cells in order to colonize and resist flushing. So the pili, as we saw, are thin protein tubes that originate from the cytoplasmic membrane, and they're found in virtually all gram-negative bacteria, but very rarely in gram-positive bacteria. So this is an adherent structure for most gram-negative bacteria. Now remember, the pilus has a shaft composed of a protein called pilin, but at the end of the shaft is an adhesive tip structure, a sequence of amino acids that has a shape corresponding to the shape of specific glycoprotein or glycolipid receptors on our cells, as we see in Figure 1. So again, here we see the shaft of the pilus in yellow, and then the adhesive tip at the end that has a shape that fits a specific receptor on a specific type of host cell. Now, there's also pretty good evidence that the binding of pili to a host cell can serve as a trigger for then activating the synthesis of some cell wall adhesins. So often the pili play a role in adhesin production as well. And this is an animation we showed you back in Unit 1, how most gram-negatives first use pili and then cell wall adhesins to adhere to receptors on the surface of host cells. This represents Neisseria gonorrhoeae that uses both type 4 pili and adhesins to attach. So let's take a look at this animation. So in this case, we see that as the bacterium gets down near the surface of a cell, it has this pilus sticking out with the adhesive tip on the end, kind of like casting out a rigid fishing line with a hook on the end that binds to receptors. And one of the things we think the pili might do is, because bacteria have a slight negative charge and the host cell has a slight negative charge, there's normally an electrostatic repulsion between the two cells. But the pili are long enough that they're beyond that range of electrostatic repulsion. So the pili often allow a way for the bacteria to first kind of loosely attach to host cells. So here we see, as the bacterium gets closer to a cell, in some bacteria this might be through motility, it might be through moving fluids, it might be through Brownian motion, but there we see the adhesive tip lock into a specific receptor that fits that adhesive tip on the surface of the host cell. And that allows for initial attachment of the bacterium. Now, if it's a type 4 pilus, as we'll mention in a minute, remember they can allow the bacteria then crawl over the surface of the host cell and spread out over the surface and find new places to attach and colonize. But eventually the pilus depolymerizes, and as it depolymerizes, as the shaft depolymerizes, that reels the bacterium in towards the surface where cell wall adhesins can now more intimately bind to adhesin receptors on the surface of the host cell. So here we see depolymerization of the shaft of the pilus, pulling the bacterium down to where cell wall adhesins can now bind more tightly. And at that point, the organism is firmly attached to the cell and can resist flushing. So many gram-negative bacteria use both pili and then cell wall adhesins to adhere. And we'll review the adhesins in just a minute. This little animation from Howard Hughes Medical Institute that kind of shows how E. coli does some tricky things to be able to adhere to intestinal epithelial cells. So let's play this animation. So what we're seeing thus far is that the bacterium is using its flagella to swim through the mucus, as we mentioned in our previous SoftChalk lesson. And in the case of E. coli adhering to epithelial cells, as it approaches the surface of the cell, the microvilli tend to disappear or be pulled in, and that provides a nice flat surface on which the E. coli can then adhere to the cell. And again, it can use pili, not showing here, in adhesins for attachment. Now, the microvilli that we see here, those are extensions of the epithelial cells that provides great surface area for absorbing nutrients, but they also get in the way of the bacterium adhering. So through some mechanism that isn't fully understood, the microvilli tend to disappear, as seen in electron micrographs, allowing the E. coli to eventually adhere to the surface of the cell with pili and or adhesins. Let's follow this on some more. So it's now injecting molecules into the host cell with the type 3 secretion system. So what we saw there is that the adhesins, I'm sorry, not the adhesins, the molecules that are secreted by the type 3 secretion system, the effector molecules, become modified inside the host cell, and then they insert themselves into the membrane of our host cell. And that's going to eventually provide receptors for the adhesins. So in this case, what the bacterium is doing by injecting molecules with its type 3 secretion system is causing the host cell to have receptors on its surface of bacterial origin that the bacterial adhesins can attach to. So we think as the tube retracts from the type 3 secretion system, that pulls the bacterium down so that the adhesins in blue here on the surface of the bacterium can bind to adhesin receptors. So initially at the beginning, we didn't see the pili or adhesins or any of that because the E. coli is actually going to cause the host cell to develop adhesin receptors for the adhesins due to using its type 3 secretion system. So this is polymerization of actin. So that shows the role of pili and an introduction to adhesins, which is the next topic we'll look at in just a minute here, since gram-negatives often use pili and adhesins. Now remember, bacteria constantly lose and reform pili as they grow, and the same bacterium has multiple alleles for adhesive tips. It can switch the adhesive tips of the pili in order to adhere to different types of host cells and in order to evade immune defenses such as antibodies made against the adhesive tip of a pilus to prevent attachment. For example, the E. coli that cause urinary tract infections can make over 30 different types of pili adhesive tips. So when it's making one type of adhesive tip, like this round one we see here, that would allow it to bind to receptors on this particular cell in the body. But another of that same organism may be making a different adhesive tip, allowing it to bind to a different adhesive tip on an alternate host cell. So by being able to change the adhesive tips, that gives the bacteria to adhere to a variety of different host cells with different adhesin receptors. And we also talked about the type 4 pili back in Unit 1 that not only allow for attachment, but enable a twitching motility. So these are typically located at the poles of bacteria when they're bacilli, and they allow the bacteria gliding motility along a solid surface, such as a host cell. And by extension and retraction of the pili, this allows the bacteria to drag itself along the solid surface. Some of them can also have a slingshotting type motion that we mentioned, but the most common one is this just extension and retraction and dragging motion. Now, for example, here's an animation we showed you of the twitching motility, where the bacterium can produce pili at a pole. And then once the adhesive tips attach to a receptor, the pilus depolymerizes. As it depolymerizes, it pulls the bacterium towards where it's adhering, allowing it to crawl over the surface. And then it makes new pili, and they extend, and they attach, and then they retract, and it keeps dragging the bacterium along the surface so it can spread out over the cellular surface and find more places to attach. And this is a movie we saw showing the twitching motility of Pseudomonas. This area on the right is a blow-up of the area in the box. We can't see the pili here because they're too small to be seen at this magnification, but we can see the dragging motility over the solid surface here. And if it wants to go in a different direction, it just makes pili at the other pole, and that drags it in the opposite direction. But we can see over time the bacteria crawling along the surface by extending and retracting the pili. So a lot of gram-negatives that are medically important have these type 4 pili that not only allow them to attach, but allow them to spread out over a cellular surface. And there's some other little movies you showed you of the pili actually extending and retracting that you can look at if you want. So looking at a few examples, Neisseria gonorrhoeae, of course, is the bacterium that causes gonorrhea. And that first has to colonize mucosal surfaces composed of columnar epithelial cells. So pili allow for the initial binding. And the fact that, of course, Neisseria gonorrhoeae, like many bacteria, can rapidly lose and synthesize new pili with different adhesive tips, that enables the bacterium to adhere to a variety of tissues and cells in the body. For example, that allows it to adhere to the epithelial, some adhesive tips allow them to adhere to the epithelial cells of the mucous membrane. When Neisseria gonorrhoeae is typically infecting the urethra, as we see in typical gonorrhea, those Neisseria gonorrhoeae that have adhesive tips that fit the epithelial cells of the mucous membranes of the urethra can adhere there. And that allows them not to be flushed out by urine. But also through oral sex, Neisseria gonorrhoeae can get into the throat, and a different adhesive tip is able to adhere to the buccal mucosa in the throat and allow it to colonize there, causing basically a sore throat. And when it gets into the rectum, of course, it can get the rectum through anal sex, but also in women, when they're using proper hygiene and wiping properly after urination front to back so you don't put fecal bacteria in the urethra, if they have gonorrhea in the urethrum, that then allows it to infect the rectum. So about 60% of women who have gonorrhea also have the Neisseria gonorrhoeae growing in the rectum. And again, that would be a different adhesive tip that can adhere to the epithelial cells in the rectum. If a baby passes out the birth canal of an infected mother, or the gonococcus gets on the fingers and the person rubs their eyes, that can infect the conjunctiva of the eyes. And also there's adhesive tips that can adhere to sperm. And so in this case, the gonococci adhere to sperm and hitch a ride, and the sperm can then carry the bacterium up to the cervix and enter the fallopian tubes or uterine tubes, where it can lead to pelvic inflammatory disease. So by being able to make multiple different adhesive tips, having multiple alleles for different adhesive tips, it allows it to be able to infect a variety of different types of epithelial cells in the body. And then as the pili retract, subsequently then adhesins called OPA proteins allowed to adhere. And again, there are multiple alleles for that. The pili of Neisseria meningitidis, the bacterium that causes meningococcal meningitis, initially allows it to adhere to the mucosal epithelial cells in the nose and throat, where it's often asymptomatic. So maybe 85% of the people who have their nose and throat colonized, it is asymptomatic, although they can potentially transmit that to others via the nose and throat. But in about 15% of the people who have the Neisseria meningitidis colonizing the nasal pharynx, it can enter the blood, where it can cause septicemia, and then cross the blood-brain barrier, where it can cause meningitis. And we think the type 4 pili play an important role in their twitching motility to help actually cross the blood-brain barrier. And that's our highlighted bacterium for this SoftChalk lesson, Neisseria meningitidis, that causes meningococcal meningitis. Uropathogenic strains of E. coli, the ones that cause urinary tract infections, produce pili and enable the bacteria to adhere to the urinary epithelium and cause urinary tract infections. Vibrio cholera uses pili to adhere to intestinal cells of the mucosa to resist the flushing action of diarrhea. Pseudomonas is initially able to colonize wounds or the lungs by means of pili and then cell wall adhesins. So again, if the bacterium has no way to adhere to cells, let's take the case of a gonococcus. If it didn't have pili or adhesins and it got into the urethra, urine would flush the bacteria out. But if the bacteria is able to adhere by pili and then eventually cell wall adhesins, that allows it to bind tightly to the cells and resist the flushing actions of the urine, causing inflammation of the urethra or urethritis. Now, the second factor that bacteria use to adhere that we mentioned in Unit 1 are cell wall adhesins, surface proteins found in the cell wall that bind to receptor molecules on the surface of host cells and again allow them to adhere more intimately to that cell in order to colonize and resist physical removal. So many, if not most bacteria, produce one or more adhesins to colonize cells. And just like the bacteria may have multiple alleles to produce different adhesive tips of pili, they often have multiple alleles to produce different cell wall adhesins. So in figure 5, we see an adhesin, a surface protein on the cell wall, binding to adhesive receptors on a host cell membrane. And again, by being able to adhere, that enables them to resist flushing. And there's a lot of examples of this. Streptococcus pyogenes, the group A beta strep that cause strep throat, has a variety of adhesins. It produces protein F that binds to fibronectin. And fibronectin is a common protein on epithelial cells. So this way it can adhere to the lymphatics and the mucous membranes of the upper respiratory tract in order to cause streptococcal pharyngitis or strep throat. One of its cell wall components, lipoteichoic acid, is also able to bind to fibronectin and act as an adhesin. And then another protein called M protein is a third adhesin that it has. And the tips of the spirochete Treponema pallidum that causes syphilis has an adhesin that allows the tip of the spirochete to also bind to fibronectin on epithelial cells. So as we saw when we talked about motility, when a spirochete either penetrates through a cell or passes between endothelial cells and capillaries, first it starts with the adhesive tip attaching with an adhesin binding to fibronectin, and that acts like a starting point for the drilling activity, and then through the corkscrewing motility, it's able to drill through the cells and enter the blood work and disseminate to other parts of the body. The spirochete also produces an invasin that we'll talk about later on that kind of softens up the phospholipid membrane of the endothelial cells to make it easier to pass through the cell with its corkscrewing motility. So again, we see the corkscrewing motility starting out with an adhesin binding to fibronectin, and then the spirochete drilling itself through the cell or passing between the endothelial cells. And then the blood can carry it to another side of the body where it can set up an infection at a different location by using the same mechanism to leave the capillaries and enter new tissues to start up a different stage of the infection. And this is a nice scanning electron micrograph we showed you of Treponema pallidum adhering to a host cell by the tip and in the process of drilling its way through. So here's one that just started entering the cell, and here's one that's pretty much all the way into the cell. Likewise, the tip of the spirochete Borrelia burgdorferi that causes Lyme disease that has adhesins that bind to various host cells in the body. And these spirochetes, like syphilis and Lyme disease, often come in stages as they spread via the blood through their motility and their adhesives and such to start up different stages at different locations in the body. Another bacterium that kind of forms a pedestal, well, this is the E. coli actually we kind of talked about earlier, showed in the movie there, but again, we saw how E. coli can use a type 3 secretion system to inject molecules into the epithelial cell that actually become modified and insert into the membrane of the cell to reproduce a receptor for the cell wall adhesin. And then through polymerization of actin, a little pedestal forms, and that allows the bacterium a nice firm place to live and grow on the cell. And we saw that in the Howard Hughes Medical Institute video just a few minutes ago. Likewise, Helicobacteria pylori uses a secretion system, in this case a type 4, to deliver effector protein that induce host cells to display receptors for adhesins. So often the bacterium itself uses a secretion system to inject molecules into the cell that become modified, insert into the membrane of that cell, allowing a receptor to be on the surface that the adhesins of the bacteria can use to attach tightly. Bordetella pertussis that causes whooping cough produces a number of adhesins. Filamentous hemagglutinin allows the bacterium to adhere to galactose on the membrane of ciliated epithelial cells. Pertussis toxin, which leads to some of the symptoms of whooping cough, also functions as an adhesin. Produces an adhesin called protactin that allows it to adhere. So again, the whooping cough bacterium, Bordetella pertussis, produces several adhesins that allow it to bind to adhesin receptors on the host cell and resist flushing. The adhesin proteins for Neisseria gonorrhoeae are called OPA proteins, and they again allow the bacterium to bind more tightly to the host cell after it first adheres with pili. And again, there are multiple alleles for these cell wall adhesins or OPA proteins that allow the bacterium to adhere to a variety of different host cells, just like it can make a variety of different adhesive tips for its pilus. Staphylococcus aureus that causes a lot of wound and postoperative wound infections, typically causing pus-filled lesions called abscesses, produces an adhesin called protein A that allows it to adhere to a variety of host cells and also helps it to resist phagocytosis. So again, quite a few pathogenic bacteria use adhesins eventually to adhere to host cells. And of course, the third way bacteria adhere is by producing biofilms. So many bacteria produce a capillary polysaccharide matrix or glycocalyx that initially allows bacteria to adhere, but then as quorum sensing genes are activated, that leads to massive secretion of this polysaccharide matrix, forming a biofilm on the host tissue. And remember, the biofilms are where groups of bacteria are attached to the surface and are enclosed in a common secreted adhesive matrix, a massive amount of secreted glycocalyx, which is typically polysaccharide. And so many pathogenic bacteria, as well as microbiota and many environmental bacteria, actually live in these biofilm communities. And by living as a community within a biofilm, that offers several advantages to bacteria. They're more resistant to attack by antibiotics because the antibiotics don't diffuse well through the biofilm. And many of the bacteria growing in the biofilm are actually growing quite slowly, so they're less affected by the antibiotic than rapidly growing bacteria. The biofilm also traps nutrients often released by host cells once the bacteria produce enzymes and toxins, and those nutrients allow the bacteria to grow in the biofilm and allow the biofilm to mature and mushroom out. Of course, they're adhering to the environmental surface by the biofilm, so they're resisting flushing. They're still communicating through quorum sensing. And of course, they can resist phagocytosis and attack by the complement proteins inside this massive biofilm. So these are some pictures we showed you of Haemophilus influenzae producing a biofilm. Let's go back on that. I think that one we might have to right click on. Let's try that. So here's some of the pictures we showed you of Haemophilus influenzae, a bacillus-shaped bacterium, and you see the secreted biofilm sticking to the bacteria. And as the biofilm matures, water channels begin to form to deliver nutrients and oxygen, remove waste, and eventually the major water channels develop into smaller water channels, so the whole community of bacteria get fed and receive oxygen if they need it, get rid of waste products and such. And this is a biofilm of Staphylococcus aureus growing on the luminal surface, that is the inside surface of an indwelling catheter. So you see the grape-like clusters of staph surrounded in this biofilm adhering to the wall of the indwelling catheter. I'm going to reload this just quickly because the magnification went out, increased when we went to those clicks there. So excuse me while I get back to that. Okay, back to where we left off. Now, another good example of biofilm production is the formation of plaque on teeth from the microbiota in the mouth. So bacteria like Streptococcus mutans and Streptococcus sabrinus are two bacteria that are implicated in initiating dental caries or cavities. Now, these bacteria have enzymes that break down sucrose, table sugar, into its two monosaccharides, glucose and fructose, and they can eventually convert the fructose into more glucose. The Streptococcus mutans can now use an enzyme called dextransucrase that converts sucrose into a sticky polysaccharide called dextran. And it's this dextran made by the bacterium from the sucrose that forms the biofilm that enables the bacteria to adhere to the enamel of the tooth and initiate plaque formation on the teeth. So this dextran mesh traps the Streptococcus mutans and the Streptococcus sabrinus along with other bacteria and debris and forms the plaque on the surface of the tooth. But because the genus Streptococcus gets its energy strictly from fermentation, and acid is an end product of fermentation, the Streptococcus mutans and the Streptococcus sabrinus ferment the glucose from the sucrose to produce energy through fermentation. But as they ferment glucose, they release the waste product lactic acid. And so the lactic acid, the waste product of their fermentation of glucose, is released onto the surface of the tooth where it decalcifies the enamel and initiates the decay process, causing the actual cavity to form. So as you see, sugar doesn't cause cavities. Sugar provides the building block for the glycocalyx and biofilm. And as they break down sucrose into glucose, they ferment the glucose from the sugar and secrete the lactic acid that in turn initiates cavity production. So on this electron micrograph, we see Streptococcus growing on the enamel of a tooth. Now at this point, the quorum sensing has been activated. They're functioning individually. They're secreting a slight glycocalyx, and that's adhering to the tooth. Now, it's such a thin layer you don't see it, but it is adhering to the enamel by means of its glycocalyx. And then as more and more bacteria replicate and they start producing the dextran glycocalyx that forms the biofilm, and as other bacteria get trapped in that, you get the formation of the full-blown dental plaque as we see in this electron micrograph. So here we see plaque on the surface of the tooth with all the trapped bacteria on it. And again, as the Streptococcus mutans and Streptococcus sabrinus ferment the glucose portion of the table sugar sucrose, they release lactic acid, which then decalcifies the tooth. The enamel of the tooth begins to eat a hole in the tooth until eventually the bacteria get to the living part of the tooth where they can cause dental abscesses to appear. Another example, most children who suffer from chronic ear infection, which is called otitis media. Otitis media is a middle ear infection, have a biofilm of bacteria in the middle ear. And that biofilm typically contains bacteria like Streptococcus pneumoniae, the pneumococcus, Haemophilus influenzae, and Moraxella catarrhalis. And so this biofilm of bacteria chronically colonize the middle ear and of course resist body defenses and resist antibiotics. And so as a result, although the number of bacteria can be reduced and the symptoms may disappear, the ear infection then often recurs because the biofilm is still there in the middle ear. Planktonic Pseudomonas, as we saw, uses its polar flagellum to move through the mucus to make contact with surface and mucous membranes, but then it can use pilline cell wall adhesins to adhere to epithelial cells. And that then once the bacterium is able to colonize by functioning as individual bacterium, as the bacteria multiply and quorum sensing genes are activated, then this eventually enables the bacteria to function as a population where they start synthesizing a biofilm composed of a compound called alginate. And at this point then in the biofilm, the bacteria lose their flagella, become non-motile, secrete enzymes that enable the population to obtain nutrients from host cells. And eventually, as the nutrients are supplied, the biofilm mushrooms up and develops water channels to deliver water nutrients to all the bacteria. And as we finally saw, as the biofilm begins to get too overcrowded, quorum sensing enables some of the Pseudomonas to again produce flagella, escape the biofilm, and colonize a new location. So we think that many chronic and difficult to treat infections are caused by bacterial biofilms. Within biofilms, the bacteria grow more slowly. They exhibit different gene expression than the free planktonic bacteria that function as individual bacteria rather than those in a population. They're more resistant to antimicrobial agents because of the reduced ability of the chemicals to penetrate the biofilm. And so in diseases like tuberculosis, kidney stones, staph infections, Legionnaire's disease, periodontal disease, it's thought that biofilms play a major role in this. It's been estimated that as many as 10 million people a year in the United States may develop biofilm-associated infections as a result of invasive medical procedures and surgical implants. So again, one of the dangers of invasive medical procedures or putting in surgical implants is that microbiota from the skin can get in there during the process and they can form a biofilm on the implant and lead to surgical wound infections. So that is the role of bacterial adherence in promoting colonization by being able to adhere to host cells using pili with adhesive tips and or cell wall adhesins, and through a glycocalyx or biofilm, they are able to colonize host cells and resist physical flushing. And once again, you have your self quiz. You can do it then there to quiz yourself.